Friends, welcome to Biology Made Easy. Today in this video, we'll discuss about mitochondria. So mitochondria is plural, in singular it will be mitochondrion. So regarding mitochondria we'll discuss. So mitochondria is known as the powerhouse of the cell. It is present in almost all eukaryotic cells where there is aerobic respiration. But in a mature RBC, they are usually absent. The mitochondria were first observed by Colliker. Colliker in 1850, he first observed the mitochondria inside the cell, and Fleming called these structures as phyla, while Altman called this structure as bioblast, and ultimately the name mitochondria was given by Panda, and that name is continuing. So in this way, the Colliker first observed, and ultimately the name was given by Panda. Usually, if we want to study the structure of mitochondria, it is commonly observable in ordinary compound microscope. But only to study the detail of the internal structure, then we have to go for the help of electron microscope. And when to study one organelle, usually it is to be stained. And during staining, usually the cell dies. But to study the structure of mitochondria within a living cell, we can use one vital stain that is known as Janus green B. Janus green B, this is a vital stain. And if this will be used, then the cell will be living and we can study the detailed structure of the mitochondria. Now, if we discuss the size, shape and number. So, the size usually it varies from 0.1 to 1 millimicron in diameter, and usually 1 to 4.1 micrometer in length. This is the average, or it ranges in a length, and this is in the diameter. This is normal one. Now coming to the shape, usually the mitochondria are sausage shaped. Sausage shaped, and sausage shaped means they are just like plate like. But in addition to this, they may be also cylindrical, or they may be filamentous, or sometimes they may be tennis racket shaped. These are the different sizes, but this sausage shaped, the plate-like structure is very common. Now coming to the number of chromosome, number of mitochondria per cell. All the mitochondria, whatever number it is present, all the mitochondria that is present in a cell is known as chondriome. And usually the animal cells have more mitochondria than the plant cells, because in plant cells most of the activities carried out in the chloroplast where the ATP is generated, but in animals such structures are not there. So animals have more mitochondria than plants. Now what is the number? There is only a single mitochondria per cell in case of chlorella and also microasteris. While there will be around 25 mitochondria in case of sperm cells, and around 500 to 1000 per cell in case of liver cells, and around 30,000 mitochondria per cell usually in some oocytes, and the maximum number of mitochondria that is present in case of flight muscle cells, and it is around 5 lakhs. So these are usually present in the flight muscle cells of the birds. And where this mitochondria will be more located, they will be more located where it requires more energy. The cells which are more active, they require more energy, so naturally they require more amount of mitochondria. So this is about the size, shape and number. Now if we discuss the structure of mitochondria, as already we have discussed that is successed mostly, and mitochondria consist of two membranes and two chambers or two compartments. So there is one outer membrane. This is the outer membrane. The outer membrane is smooth. We'll discuss one by one. Then there is the inner membrane is there. Then the inner membrane has several enfoldings like this. Like this. So this is the outer membrane. This is the outer compartment or outer chamber. This is the inner membrane and this is the inner compartment or inner chamber. That means two membranes and two chambers or two compartments. Now we'll discuss the outer membrane. So when we'll discuss the outer membrane, as you see, it is quite smooth, and it is permeable to most of the metabolites because it contains a special type of protein known as porins. So it is permeable to metabolites, and it contains special type of protein that is known as porins. So that is why it is more permeable to metabolites. This is the outer membrane and it is smooth as you can see. Now the next one is the inner membrane. Now the inner membrane, it is not smooth and it has several enfoldings, and the enfoldings are known as cristae. That means it has not smooth. It has several enfoldings, and the enfoldings are called cristae. So now when there will be more number of cristae and when there will be less number of cristae, it depends upon how much active mitochondria is and how much of energy it is producing. If it produces more amount of energy, then the cristae will be like this, and that is in that case it is known as the condensed state. Condensed state means there is a production of more ATP, condensed state, or it may be in the orthodox state. Orthodox state means no ATP production. So in that case, the cristae are not well developed, and that actually this structure of the mitochondria is changing. It has been observed by the attaching video to the microscope. When it is more active, it is condensed state and the cristae are well developed, and when there is no much production of ATP, then the cristae are not well developed, that is the orthodox state. That means this is changeable, both orthodox and condensed state is changeable depending upon the activity of. Now, the inner membrane, which is has several enfoldings and cristae, they also contain more amount of proteins and enzymes. They contain more proteins and enzymes, and it contains a special type of double phospholipid. It contains a double phospholipid which is exclusive only to the inner membrane, that is known as cardiolipin. That means instead of 3 phosphoric acid there are 4 phosphoric acid. That is why it is known as double phospholipid, and it doesn't allow the movement of all the ions into the through this inner membrane. That means it is selectively permeable. The inner membrane is selectively permeable. So, if now you will compare both the outer and inner membranes, the outer membrane is smooth, this is not smooth because it has foldings. The outer membrane is permeable, this is selectively permeable. The outer membrane has less proteins, but in case of inner membrane, there are more proteins and enzymes. That means both inner and outer membranes of mitochondria are structurally and functionally different. This is an important question. Both of them are structurally and functionally different because they contain different amounts, they are differently permeable, and they contain different types of proteins and different the permeability, that all varies. Now, on the inner side of the inner membrane, that means in the cristae, there are some pinhead-like structures known as oxysomes or F0F1 particles, or also they are known as Fernandez-Moran particles. So on the inner side of this, there is some pinhead-like structures like this, and they are present throughout. Throughout they are present like this, and these are known as elementary particles. Or they are known as oxysomes, or they are known as F0, F1 particles. Also they are known as Fernandez-Moran particles, according to the name of the scientist, Fernandez-Moran particles. And what is their number per cell? Usually the number per cell varies from 10 to the power 4 to 10 to the power 5. In a particular mitochondria, that large number is there. And each oxysome or elementary particle has one base and one stalk and one head. And in this head part, the ATPase enzyme is there, and this is these elementary particles are responsible for synthesis of ATP during oxidative phosphorylation. So that we will discuss while discussing the function, and also how it is produced in respiration, and in case of respiration the details are discussed. So this is the structure of the inner membrane, which is completely different from that of the outer membrane. Then the third part, the third one, now these are the two membranes. Now then outer chamber and inner chamber is there. Now the third one is the outer chamber. Now this is the outer chamber. And this outer chamber, it is usually its width is 60 to 100 angstrom, and this is also known as perimitochondrial space. This is known as the outer chamber is also known as perimitochondrial space, whose width is 60 to 100 angstrom. And in this part there is some fluid matrix, and some enzymes are there. But when during respiration, when the electron transport system takes place, this outer chamber becomes highly acidic due to accumulation of protons. During that, here the accumulation of protons are there, and it becomes highly acidic when there is electron transport system and synthesis of ATP. So this is regarding the outer chamber. Now the fourth one is this inner chamber. The inner chamber is also known as a matrix. So the fourth is the inner chamber. Inner chamber, or this is known as matrix. Now in the inner chamber there is a semi-fluid. Semi-fluid matrix, and this the inner chamber, it contains all the enzymes of Krebs cycle except succinic dehydrogenase. So it contains all enzymes of Krebs cycle except succinic dehydrogenase. Except succinic dehydrogenase. So this is the exception. Except that all the enzymes of Krebs cycle are present, except this succinic dehydrogenase, which is not present in the matrix, that is attached to the inner membrane of the mitochondria, where the electron transport system takes place. In addition to this, all these enzymes and the fluid matrix, it also contains also this mitochondrial DNA, which is circular and naked, means no histone proteins. It also contains 70s and 50s ribosomes. Usually in case of human beings, mitochondria it is 55 years, in other cases it is 70 years, and this is circular. And this is the mitochondria, the DNA is circular and naked, while in case of the human mitochondria, it is the ribosomes are 55 base, and in the other case 70 years. And in addition to that, also different types of RNAs are there. Now, in addition to that, also you will get some vitamins like riboflavin and vitamin E. That is riboflavin, and riboflavin is yellow in color. That is why mitochondria matrix looks yellow in color. Then there is vitamin E, and several minerals are also present in traces, like the manganese, calcium, phosphorus. These are the different types of traces in the present. That means this is the chemical constitution. That means it consists of proteins, lipids, DNA, RNA, ribosomes, and vitamins, as well as the different minerals in traces. And all these things are present within the mitochondrial matrix, where the TCA cycle or Krebs cycle takes place.